I thought I would do a really short recap on what I covered last something, like I think it was like December 12th or 13th or something like that. We covered the mini project. The goal in the mini project is to create a new chromoprotein and submit that new chromoprotein to the registry. If you don't know, chromoproteins emit color in ambient light, so it's an instrument-free type of diagnosis rather than fluorescence proteins that you have to use a UV light for. You can tell that these have been properly transformed in regular light. The mini project is split into three main components. The first of the components is the planning step, is where you create the outline. I know, Austin, you have been working on that a little bit. The outline, I did a big in detailed description of how you should create your outline in that video, which is right here. You can click on it if you want. Next is wet lab, which takes typically the longest. It's where you're using your outline and also your lab notebook that you're going to be typing up after every or before and after every lab visit. You're going to start putting those to use. So you're going to come to lab, you're going to start doing the procedures and protocols. You're going to start weighing out and figuring how much each material that you need is. And that's where you're going to learn most of the hands-on stuff and actually start cementing that knowledge that you learned in the planning step. Finally, there's the diagnosing and submitting step. Here, you're going to be either diagnosing what went wrong with your project or submitting the fully completed new chromoprotein part. You'll do a short write-up there with regards to which one you end up falling into, though it's not, don't take it too seriously. My main goal is to get you guys through mini projects so we can finally get independent lab members working on our main project, pathogen predictor. Okay, so if you're not sure about all of this, go ahead, check that presentation, which is linked in the lab, lab resources channel. There's also that video that's attached to the iGEM playlist now, and that goes into in-depth detailing with regards to the mini project. Okay, so this is something new I'm starting this semester. It's like extended concepts, so I'm going into things that go past the general synthetic biology knowledge that you should know when you're in iGEM. And our first concept is with regards to our project. So we're covering quorum sensing in as much detail as I can wrap my hand around. So quorum sensing in a nutshell is the regulation of gene expression based on population density. So some bacteria always have these genes, right? And they code them, but not all the genes are coded at once. So they want to regulate these genes based upon how many other bacteria are present or are next to them in order to make the thing that they're coding for ever more prevalent. So it's very, very useful with regards to a few things. There's actually more than just three, but I thought I would include these. The first is symbiosis. So rhizobia, they are into fixing bacteria that help with plants. They have a symbiotic relationship where they fix the nitrogen for the plants, and it provides a living environment and an eating environment for them. Next is virulence, which is what's, I guess, better known. V. cholerae is probably one of the most well-known bacteria out there. It causes cholera. And when these bacteria get together and their quorum sensing starts enacting, they start producing cholera-toxin, which causes most of the diarrhea and the side effects from cholera. And also biofilm formation. Probably the most common form of biofilm is plaque formation on your teeth. And that's actually a quorum sensing between over 700 different species. So these species talk to each other through these autoinducer or peptide molecules. They're called signaling molecules, which you can see right here. And they secrete them, and then each of these bacteria start taking them up, and they start enacting a response to an increase of those signaling molecules. It is worthy of note that it is not just a intraspecies set of molecules, though. Some of these molecules work in between hundreds of species. Okay, so I thought I would attach a little diagram of how this works. This is an abstracted version of quorum sensing, so it's pretty high level. It starts where you just have a few cells, very, very low density with regards to cellular density. So these cells are secreting these signaling molecules. There's just a few surrounding them. And most of the time, these signaling molecules, because there's not enough of them, because there's not enough cells secreting them, they're just going to diffuse far, far away from these cells, so they can't reuptake them. So that's kind of pointless. But once these bacteria start replicating more and more and more, and the density starts increasing, each one of these is going to start producing these AI2 or just signaling molecules. Because of that, the concentration of these molecules is going to increase by a lot. And once it hits a certain stage, these cells are going to start uptaking them, and it starts enacting the quorum sensing operon. So you're going to start enacting those genes that the quorum sensing codes for. One of those genes could be biofilm formation. So once it reaches high density, it's going to tell all these bacteria to start forming a biofilm. And they form that biofilm in order to protect them against external hazards, or just to reproduce better through horizontal gene transfer, stuff like that. A closer look, there's a whole bunch of, it's a very, very complicated process. I'll get into it a little bit later. But once it diffuses out, it goes into another bacteria, which it attaches to a protein or an enzyme, and then that starts enacting the operon, right? Of note, though, this is actually like a positive feedback loop. So once these absorb the autoinducer molecule, it actually starts transcribing the production of more autoinducer or signaling molecules. And so you can see how this can get pretty crazy in a short amount of time. So yeah, 1 comes in and 3 comes out, starts mass producing them, and the higher the density becomes, the more that gets produced. Okay, so it is worthy to note that different types of bacteria use different types of quorum sensing. So gram-positive bacteria use a specific signaling molecule called autoinducing peptides. These are just pretty much proteins. And then gram-negative bacteria use a signaling molecule called autoinducers. Of note, the gram-negative are more important to us because it's the bacteria that we work with in lab, and also the bacteria that we're looking for our project, which is Salmonella. So gram-negative bacteria use a specific operon or a specific system called the LuxI. I think you can replace the I with an S. I'm not too sure about that. I've seen LuxI and LuxS code for around the same things, but the LuxI, LuxR type quorum sensing systems. And LuxI and LuxR is great interest to us because we're trying to detect gram-negative bacteria in the local environment, and specifically in food manufacturing. I'll get into these operons in a minute. So the molecule that we're going to be using, which is specific to gram-negative signaling molecules, so the autoinducers, is going to be autoinducer 2. So there's of course autoinducer 1, but we're using autoinducer 2 because of its ability to transfer across different species. So it's a very, very common type of autoinducer. Because it's so common, we can use it to detect Salmonella, even if we're using E. coli to do so. So what we're trying to do here is we're going to try to make a culture of bacteria with the sensing capabilities to detect the concentrations of AI2 that's secreted by Salmonella. And that's our project in a nutshell. This is a picture of autoinducer 2. It's not very informative, but I thought I would just include it to increase this slide's depth. Okay, so this is the actual AI2 pathway. It's very, very complicated. I think the first time I saw this, I said, "What the hell?" So what happens is this is your E. coli cell. These are the signaling molecules, so AI2, and this is a pretty high level version of this pathway. But pretty much what happens is at step 1, these proteins that are suspended in the outer membrane, they are transport proteins, and they start importing autoinducer 2 into the cytoplasm. And within the cytoplasm, there are enzymes that are, it's also known as LSRK. Pretty much it's just, I think it's called kinase. It's an enzyme that transforms autoinducer 2 to phosphorylated autoinducer 2. So it attaches a phosphate through ATP and ADP, all that sort of stuff. And then so you can see that here, it's a signaling molecule with the phosphate attached to it. And at part 2, or I guess that was part 2, at part 3, LSR, which is a repressor. I guess I'll get into that. So LSR, because it is a repressor, it represses the transcription of all of these genes right here. And so when there's no phosphorylated AI2 present, all these genes are repressed, And so it doesn't like code or transcribe any of them. The moment phosphorylated AI2 gets added to the cytoplasm, it binds to LSR, thereby changing its structure and function, and in turn, it derepresses. So it derepresses and and the cell can start transcribing all of these different genes. Of note, this gene actually codes for LSR, so it creates more of the repressor. And this gene codes for more of LSRK, which increases the amount of phosphorylated AI2. This right here is a bidirectional promoter. So this LSR turns on the bidirectional promoter, which in turn transcribes the LSR and the LSRK, and also this humongous operon right here. Note that the LSRA, C, D, B, I think that's it, maybe D, yeah. They're all located, they code for the proteins within the cellular membrane. And so that in turn, because the derepression of this increases the concentration of these proteins by transcription, it actually increases the amount of AI2 the cell can uptake. So because more of these proteins are being transcribed, going to the cellular membrane and starting to let more AI2 in. So that's pretty much AI2 in a nutshell. It's pretty complicated. Two things to note here. Lux S, which I also think might be Lux I. I need to do a little bit more research in that department. It creates AI2. So in our project, when we're trying to detect the concentrations of Salmonella, we do not want our E. coli cells to produce AI2 because that in turn would turn on our system, which would make it, buggy. It would start enacting on itself. So we want to somehow either mutate this out of the operon. I think there were a few iGEM teams that did that in the past, or we need to do a deletion in order to remove this gene from the E. coli. So also, I think I already said this, LSR represses both operons, so it represses this and this. So this is pretty much the same slide. I just wanted to ask you guys a question. We want to insert a gene that increases or creates fluorescence within the E. coli when it comes into sufficient contact with external AI2. At which point or where do you think we should insert this gene that codes for the fluorescence? And this is a question any of you guys can answer this. Okay, no response. So I guess I'll explain it. Because this is the promoter and it gets derepressed by the phosphorylated AI2. Once this gets derepressed, these starts being transcribed. So the best place to put our gene of interest is either right here or right here. I saw most iGEM teams typically put it right around here next to the LSRA gene. And if it is inserted right there, then once this transformed E. coli comes in contact with AI2, it will thereby depress this, turn on transcription of this operon, and then start transcribing your fluorescence gene. In short, in a less complicated manner, E. coli comes in contact with signaling molecule, starts fluorescing. That's pretty much it.